Yeah, the scientific procedure was multifaceted. I mean, I had a good-sized lab, and so we had divided people up into different primary goals, and would meet daily. And the first goal was, if a patient has fully developed AIDS, forget risk groups at the time. We didn't want to get into the risk group. You know, they had AIDS, documented AIDS. Could we take their blood, get their T cells, like we would do with any, like the leukemias, like we did with the leukemias, the same protocols we did with leukemia. Take those T cells, clean them up from other cells, grow them with interleukin-2, our old friend, and see if we could find any evidence of release of virus particles, and if we were right that they would be retroviruses. And how to do that was with a surrogate marker. You don't use electron microscopy in those days, except maybe 1 or 2 pictures just to confirm or to see the structure of this particular retrovirus. The assay was reverse transcriptase.
Well, I don't know whom you interviewed, but if you don't believe me, go and ask Baltimore or Vermont, and I'm sure they will confirm that reverse transcriptase activity is a characteristic of retroviruses, but it's not specific to them.
What do you mean when you say reverse transcription is a characteristic of retroviruses, but not specific?
Oh, let me give you an example. Hair is a characteristic of humans. You know, black, white, or yellow, we all have hair, but it's not specific because there are many animals, for example, cats, dogs, which also have hair. And finding a hair in a room, it doesn't mean that a human being was there, or a cat or a dog.
Are retroviruses the only ones that can reverse transcribe?
No. There are other forms of reverse transcription that are used in various ways inside the cell. For instance, the ends of chromosomes are made by a reverse transcription process. That's how they're maintained stable. There is reverse transcription in the inheritance of all of our cells, which comes about from endogenous genetic elements in the cells, or in the cells of our ancestors. Because once that information gets into our cells, into our genomes, it stays there forever. So it could be that we've inherited information from monkeys or from other animals that are in our lineage. And so, no, reverse transcription is actually very widespread. Something like 50% of the DNA in our cells comes about by reverse transcription.
Oh, that much?
Yes.
Oh, wow. Okay. I thought it was 1 to 2%. Going back to, I thought it was 1.5 to 2%. The 50% that the retroviral DNA, or retroviral genes in our DNA, what biological function do they serve?
Now, we got to make a distinction. When I said 50%, I'm saying 50% of the DNA came about by reverse transcription. But it's not all retroviruses. Lots of it is just repeated elements that are there as what we generally consider to be parasitic DNA. DNA which is, other people call it selfish DNA. DNA which is in there because it's able to copy itself and reintegrate itself in other places. And this is something that's going on all the time. And it builds up.
Why do you think they used reverse transcription to prove the existence of a virus if they knew that it wasn't specific to viruses?
I don't know. It's as simple as this. I don't know. In fact, they went one step further. In the second experiment, they have taken the cells from their patient and mixed them with cells from a normal blood donor. And in that culture, again, they put PHA and other growth factors, substances, which make the cell to survive and grow. And again, they found reverse transcriptase activity. And this time, they claim that the finding of reverse transcriptase activity in the second culture proves that the virus was transmitted from the patient's cells to the normal donor cells. And in fact, they say that this proves isolation of HIV.
But they must have used some other criteria to state that they had a new virus?
There was no other criteria. In the first, they had a third experiment, but in the first and second experiment, that's how they are described in their paper. It was the first experiment, reverse transcriptase activity, the second experiment, reverse transcriptase activity. They are the only evidence they have published.
So in
from the first and the second experiment.
So in both experiments, they fed the culture substances which artificially cause reverse transcription.
Yes. Yes, they fed them. You know, just one of them, PHA, by itself, it will cause reverse transcription in normal cells, in non-infected cells.
What are your problems with the third experiment?
Well, the problems, first of all, again, they took the supernatant, the fluid from their second experiment, and put it in a culture which contained umbilical cord lymphocytes. And in that culture, they also found reverse transcriptase activity. Again, the reverse transcriptase activity, they already use there this PHA and other substances which can cause reverse transcription in any culture, if the conditions are right. Then we have the virus particles. They have published electromicrographs showing virus-like particles budding from the cells and released in the supernatant. But just seeing virus-like particles is no proof that they are viruses, because you can have virus-like particles and they're not viruses. In fact, the third problem with the virus, with the particle, is that they use umbilical cord lymphocytes, which originate from placenta. In placenta, it was known in the 70s that they release virus-like particle and they have reverse transcriptase activity. So find them in this culture is no proof that they originated from Montanier's patient.
In Montanier's paper, they publish electron micrographs of HIV.
No.
We see pictures of the virus.
We see pictures. These particles were in, as I said, in umbilical cord lymphocytes. And umbilical cord lymphocytes originate from placenta. And there was already a lot of evidence by this time that placenta can release virus-like particles. What's more is Montagnier, when he described the particles he has seen, he said these particles are typical type C particles. Viruses are, retroviruses are classified in different categories, one of which is type C particle. And they said that the virus they have seen is a typical type C particle. Typical type C particles have been many times published in the 1970s from normal placenta. In fact, most of the placentas was shown to have reverse transcriptase activity and typical type C particles. So finding these particles there, even if they were virus, they could have been just originating from the normal placenta. So finding the particles and finding reverse transcriptase activity in the umbilical cord lymphocyte culture does not prove that the patient was infected with a virus.
The two main things that told you had a retrovirus was the reverse transcriptase activity and the fact that the cells died.
And then we immediately call our guy who was responsible for electron microscopy and said, please, could you look under the microscope whether you can see virus particle and if it resembles to a retrovirus.
Okay.
Okay.
And what did you see? What did you guys see when you looked under the electron microscope?
And after quite, it was very difficult because it was only few cells infected, so it was a very difficult task for him to find a cells that was just producing this particle, but finally he found it. And he found one lymphocyte with a budding particle, typical of retrovirus, and very close from these cells, one complete mature particle that resembled to a retrovirus.
In that paper, he had only one electron micrograph. And the virus could be identified as sort of a retrovirus, but it could have also been an arenavirus. Now, these are specifics that are only important for experts. But when we saw that photo, we said, suggestive, but not convincing.
I've seen these publications, stamp-size images. It's a nuisance. It's a nuisance. You do not really see much. And I do not know how good electron microscopy was really done, but they probably should have done a bit more, and then they would have been very safe. And electron microscopy is an art and a science. Art and science, and nobody is able to judge what the electron microscopist is really doing. You have to have experts discussing these things. Sometimes people are far from the microscope. They do not understand structure and function. They like to have a nice image. That's all. They do not understand sometimes, and that's a danger. I could imagine if Montagnier would have pushed the structure, the fine structure of HIV a bit more, he would have been more convincing.
Would I be right in saying that from the pictures alone, though, in culture, you can't prove what virus you found yet?
Of course, electron microscopy is not sufficient to prove you have a retrovirus. That's clear. You need other characteristics like the density. The reverse transcriptase activity is the key enzyme, which is at that time was only associated with retroviruses. You know, that's how we describe it first. You know, we didn't describe the electron microscopy as a proof. We described the reverse transcriptase activity as a proof it was a retrovirus. But here also we were criticized by some people from Gallo's lab saying, are you sure you have a reverse transcriptase activity there? Could it be a cellular DNA polymerase? You know, we had this kind of question.
So Montagnier, he wanted to show not only that he had a retrovirus, but that the retrovirus was new, was not one of the two other retroviruses which Gallo claimed to have discovered at the end of the 1970s and the beginning of 1980s in leukemic patients. For this, first of all, he said he purified HIV. He obtained, by purification, we all agree, he said, and Montagnier himself admits that to prove that a virus is a new virus, then you have to show that it has proteins which were not present in any other retrovirus. And to do that, he said, you must purify the virus. He admitted that in 1997, that you must purify the virus. And in 1983, he claimed to have done exactly that, to have purified the HIV particle. Now, we may have differences as to what isolation means, but when it comes to purification, there is no disagreement. By purification, they mean to obtain particles, retrovirus particles, separate from anything else, which at least from anything else which contains proteins and RNA.
Now, when this virus is in this supernatant, it's not purified, okay? Because the cells are releasing plenty of things, not only the virus, cellular protein and so on, okay? Yes. So that means that in the supernatant, you have a mixture of everything, including the virus. Then you have to purify.
Okay.
Okay. This is the second step. Then you try to purify the virus from all this mess.